Hi guys. So as I promised, I'm recording today's lecture. Starting from next week, I will actually take suggestion from one of students, and suggestion was to turn on my Zoom while I'm talking, so I don't have to give the same lecture twice, which I think is kind of helpful. So thank you. There was a student's great suggestion. So just a brief, this is for me like a repetition of what we talked today. Lecture is reduced, and I hope you liked it. So last class we talked about phototrophy. Today we will cover chemolithotrophy and fermentations, and next week we will talk about anaerobic respiration, and we will end up with hydrocarbon metabolism. So after that we will transfer to functional diversity. So chemolithotropes are organisms that can use inorganic compounds to obtain energy for their metabolism. And from evolutionary standpoint, chemolithotropy may have been the first one form of energy conservation that evolved on Earth. So if you want to know why is that, like how do we know that these chemolithotropes were the first? Well, we know because we see that chemolithotropy is actually very widespread among those lineages that are on the bottom of phylogenetic tree, and they are present in bacteria and archaea. That means that they developed very early from common ancestor. Most chemolithotropes are also autotrophs, which means they're using carbon dioxide as a carbon source. They're capable of carbon dioxide fixation. And as you know from last class, in order to use carbon dioxide, this organism needs to have a lot of ATP and reducing power, NADHs. So chemolithotropes are able to create ATP and reducing power through oxidation of highly reduced compounds such as hydrogen sulfide or elemental sulfur. We'll talk about hydrogen and iron oxidizers. So they literally extract electrons from these compounds and use those electrons to create ATP and NADHs, because they need to invest a lot of ATPs and NADHs to fix carbon dioxide. Some of them are using Calvin cycles, some of them are using the other two cycles for carbon dioxide fixation that we talked last class. So we will cover all these energy sources: hydrogen sulfide, elemental sulfur, hydrogen, and iron. And most of the time, carbon source for chemolithotropes is CO2, inorganic carbon. And these are all indicators that chemolithotropy was the oldest metabolic process, because if you go back to our first two lectures, when we talk about what was available 4 billion years ago, so these compounds present here were available, which is the reason why bacteria learn how to use these first. Bacteria and archaea, sorry. So inorganic sources are very abundant in nature. So the reason for a huge diversity of chemolithotropes is because their energy sources are highly abundant. And we will start with hydrogen as the first energy source, and then we will end up with ammonium as the last, and then we'll talk about fermentations. So bacteria that use hydrogen as electron donors are called hydrogen bacteria, so nothing very creative. And hydrogen is known as a very good electron donor. So bacteria and archaea are able to couple oxidation of hydrogen with reduction of various electron acceptors. Some of them are nitrite, sulfate, iron 3+, not 2+, 2+ is to be oxidized, and carbon dioxide. Now, there are hydrogen oxidizing bacteria that are able to use oxygen as a terminal electron acceptor. And these aerobic hydrogen oxidizing bacteria are very specific, and they're kind of living on the interface between oxic and anoxic area, and I will mention them later on, and later in class when I cover hydrogen oxidation, then we will talk about these organisms specifically. The way how hydrogen can be oxidized by chemolithotropes is through enzyme hydrogenase, and they develop two types of hydrogenase. One is cytoplasmic, which is soluble, and the other one is membrane integrated. Now, most of hydrogen oxidizing organisms have only one hydrogenase, the one that is integrated in membrane, but some organisms have both, and soluble and integrated. So if there are both of them present, that's evolutionary advantages. Why? Because if they have membrane-bound hydrogenase, that one is involved only in energy conservation and electron transport chain. If they have soluble enzyme in cytoplasm, that one is involved in autotrophy, fixing carbon dioxide. So if you have only one type present, that one has to do double labor, and that's why the one that has species that have two are evolutionary advantages comparing to those that have only one. As I said, hydrogen oxidizing bacteria are fixing carbon dioxide via carbon cycle. They are autotrophs, but some of them can also grow as chemoorganotrophs. And what that means is that they can use organic compounds as a carbon source. So if they have a choice between glucose and hydrogen, between organic and inorganic source, they will prefer glucose because the glucose is giving more energy. So if they are given an organic source, then they will not even transcribe hydrogenase. So we call these bacteria facultative chemolithotrophs. They have mixed nature. And there is ecological significance of these hydrogen oxidizing bacteria, and here is why. So when you have oxycarriers, hydrogen production is very low. So the reasons why hydrogen is low in oxycarriers is because there is no fermentation, and hydrogen is byproduct of fermentation. So if there is no fermentation in oxycarrier and fermentation is happening in anaerobic environment, then hydrogen amount will be low. And then whatever is present of hydrogen there gets consumed rapidly by anaerobic organisms. So basically, hydrogen oxidizing bacteria must have alternative metabolism to survive. That's why they develop this ability to do chemoorganotrophy and to oxidize something organic if it's present. Now, in nature, these bacteria are shifting between chemoorganotrophic and lithotrophic style, and all depends which nutrients are available in nature. Don't mix up those facultative chemolithotrophs. They are not the same as hydrogen oxidizing bacteria that use oxygen. These hydrogen oxidizing, oxygen reducing are dominant at the interface between oxic and anoxic area. In terms of hydrogen sulfide, that is also very good reduced compound. And bacteria that are using hydrogen sulfide, other than purple and green bacteria that we covered last class in photosynthesis, so these bacteria are called colorless sulfur bacteria, and they are not the same as anoxygenic phototroph. They're quite different. They don't need light as energy. In this case, the only source of energy is hydrogen sulfide. Or they can use elemental sulfur. They usually store as granules. So they are similar to anoxygenic phototrophs, but they are different type of organisms. They can oxidize hydrogen sulfide or elemental sulfur in two modes. One mode will be through intermediary. So when hydrogen sulfide is oxidized, final product is sulfate, SO4 2-. Or they can do it without intermediaries. So though in mode 1, you will have other compounds in between that are available in full lecture, but in mode 2, it's happening without intermediates, but if organism is doing second mode, it has to be done through SOX system of genes. In terms of iron oxidation, it has to be ferrous iron, 2+, full of electrons. So these chemolithotrophic iron bacteria are able to oxidize ferrous iron, and basically at acidic pH, this reaction with iron oxidation is giving very small amount of energy. What that means is that iron bacteria must complete the oxidation of large amounts of iron just to produce small amounts of cell material. Now, when iron is oxidized spontaneously, 2+, it will form 3+, so ferrous to ferric, and this ferric iron can form hydroxides, and they precipitate in aquatic environment and lower pH, which is the reason why iron oxidation bacteria that extract energy from iron, evolved to be acidophilic. The best known bacteria are those that I really want you to know. You will recognize them by the species name ferrooxidans. They can be Acidotheobacillus or Leptospirum ferrooxidans. They're important to be known. Both of them grow autotrophically, and you will find them in acid-polluted environment. And among archaea, there is ferroplasma that's very extremely acidophilic iron oxidizer and grows at pH values that apparently can go below 0. Some of the examples of iron oxidizing bacteria other than Acidotheobacillus and Leptospirum ferrooxidants are Gallionella and Leptotrix. They are also iron oxidizers, but they are specific because they use oxygen as a terminal electron acceptor. So these bacteria live basically at the surface, and they evolved a system that enables them to rapidly consume oxygen and to rapidly oxidize iron and reduce oxygen, because iron 2 plus in the presence of oxygen spontaneously get oxidized to 3 plus, and then it's useless. So these Gallionella, Spheronitulus, and Leptotrix evolved to be able to rapidly consume iron and before it gets exposed to oxygen. And last but not least, ammonium. So ammonium, when we talk about ammonium as a chemolithotrophic source of energy, we have to know which compounds, which reduced compounds are nitrogen containing. That can be ammonia or nitrite. A nitrite is NO2 minus ammonia is NH3. So these compounds can be oxidized aerobically by chemolithotrophic nitrifying bacteria in the process that is called nitrification. So basically, when you oxidize ammonia, next step is nitrite. The other group of specialized organisms that can oxidize ammonium, so ammonia ion, are ammonia oxidized anaerobic ammonia oxidizers that are called anamox. Now, nitrifying bacteria, and in next slide I will mention specifically who they are. So they are very distributed in soils, wastewater, and oceans, and they are very significant for wastewater treatment, which we will see soon. Now, in aerobic conditions, bacteria and archaea oxidize ammonia to nitrite, so NH3 to NO2 minus. So these bacteria have an enzymatic system that is very well developed, and I will talk about that when we talk about functional diversity in lecture 10. So we shortly call them ammonia oxidizing bacteria, and there are also ammonia oxidizing archaea that also oxidize ammonia. Just apparently ammonia oxidizing archaea are more sensitive, so they grow where there is a lower level of ammonia, because they develop enzymes that have high affinity for the substrate. The full process of nitrification is completed by second group of bacteria called nitrite oxidizing bacteria, or NOB, because they convert nitrite to nitrate, NO2 minus to NO3 minus. So process of nitrification cannot be completed by only one group of organisms. It requires AOB and NOB. So nitrifying bacteria, both AOB and NOB, use Calvin cycle to fix CO2, which is very energetically expensive. Means it requires a lot of ATP and NADH, which is the reason why nitrifying bacteria run reverse electron flow to generate NADH. So because energy cost is very large, so they need to have very extensive process, nitrite oxidizing bacteria develop alternative metabolism, so they can grow as chemoorganotrophs. But AOB, unfortunately, do not have alternative metabolism. They are obligate chemolithotrophs. Nitrifying prokaryotes are very important for ecology, so they play a huge role in nitrogen cycle. When these organisms, AOA and AOB, along with NOB, they convert ammonia to nitrite, and this is key, and nitrite to nitrate, and nitrate is key nutrient for plants. Nitrifiers are also important in wastewater treatment because they can remove toxic amines, and then they release less toxic compounds. Now, in lakes, where there is decomposition of organic matter very low, where the composition of organic matter is highly extensive, ammonia will be produced, and ammonia oxidizing bacteria will oxidize ammonia to generate nitrite, and NOB will convert nitrite to nitrate. And nitrate is a great food source for algae and cyanobacteria. They prefer nitrite to grow. Now, here I probably should mention that AOB and NOB are completing nitrification together, but there is a group of organisms called COMAMOX, which stands for complete ammonia oxidation. So these organisms are doing oxidation of ammonia all the way down to nitrite. So basically like integrated AOB and NOB, but they're very hard to be cultivated in the lab. So, but they will be much better for wastewater treatment and for treatment in plants, because what's happening is, so AOB and NOB can generate a plant nutrient, nitrite, but they have to be together to do it. Now, what happens is when soil is very flooded, one example, soil becomes anaerobic, and then ammonia oxidizing bacteria have that unfortunate feature that when they are exposed to anaerobic area environment, they switch metabolism and they don't produce nitrite, they produce nitrous oxide. And nitrous oxide is greenhouse gas. That's why COMAMOX organisms that can do complete ammonia oxidation all the way to NO3 minus, they don't produce nitrous oxide, so there is no harm to the environment. That's why they would be much better, but they grow much slower and they're hard to maintain. These organisms called anamox are quite different. So unlike nitrifiers, which are strict aerobes, anamox are anaerobic organisms. They use ammonium as an electron source, but they reduce nitrite as a terminal electron acceptor. So that's why they are anaerobic. So anamox stands for anaerobic ammonia oxidation, and they are very unusual group of organisms. In this case, ammonia is donor nitrite acceptor. The product is nitrous gas, N2, but also there are other products that are very toxic. That's why they are kept in specific structures in cell. The example is bacteria Brocardia anamoxidans, and this is unusual bacteria that doesn't have peptidoglycan, and cytoplasm contain those membrane and closed compartments, which is very unusual for bacterial organisms, and those compartments are called anamoxysomes. This is unique structure, anamoxysome, present only in this organism, so it reminds on eukaryotic organelles, but it's not organelle, just reminds on it. And lipids that build anamoxysome are not typical bacterial lipids. They're called the lateral lipids, and they contain glycerol moiety and cyclobutane C4 ring. So unlike bacteria and archaea that contains fatty acid as attached to the glycerol, and this C4 ring is attached to glycerol moiety via and ester and ether bond. So a combination bacterial and archaea. Remember that bacteria evolved ester bond while archaea have ether bond in lipids. These guys have combination. So lateral lipids, very important to know as a unique structure. Why they are significant? So the source of their nitrite is coming from AOB and AOA. So when AOB and AOA oxidize ammonia, they produce nitrite, and then anamox can use that nitrite. So usually AOA and AOB coexist with anamox in those habitats that are ammonia rich, and those habitats are sewage in wastewater. But anamox often competes, can compete, actually not often, but can compete with NOBs for food source. Basically for NOB, nitrite would be energy donor, but for these anamox, its energy is electron acceptor. And it's very important, they are very important in wastewater treatment, because in wastewater you will see high levels of ammonia and amines, and they can remove this waste using ammonia and nitrite from wastewater to produce water and nitrogen gas. Again, it's very high anaerobic process, but that means it saves cost of aeration, and you don't need organic carbon source, they have ammonia. So fermentations are another another mode of chemolithotrophy. So basically fermentation is happening in oxygen-free environments where there is decomposition of organic matter. So in these environments, there will be no inorganic electron acceptors. There will be no sulfate, nitrate, or Fe ferric ions, Fe3+. So there is no inorganic acceptors of electrons. So that's why microbes have to adapt and to degrade organic compounds in metabolic process called fermentation. But because there are no electron acceptors, they cannot run electron transport chains. So how will they create ATP and NAH and the NADH? They will create it through substrate level phosphorylation or SLP. Now, in fermentation, substrate serves as both electron donor and acceptor, and ATP is synthesized through SLP, substrate level phosphorylation. There are two types of fermenters, primary and secondary fermenters. Primary fermenters ferment sugar to make acids or alcohol. You probably heard about lactic or ethanol fermentation, so those are primary products. And secondary fermenters use products of primary fermenters to generate gases such as methane, sulfide, or acetate. Acetate is not gas, but other products, carbon dioxide as well, hydrogen also. So mostly products of fermentation are excreted. A very small portion ends up in microbial biomass. And excreted products are acids or alcohol. They are end products of catabolism. Very often, hydrogen is also byproduct. You can classify fermenters by substrate that is fermented or product that is formed. And broad categories are alcoholic, lactic, mixed acid, butyric acid, or acetogenic fermentation. And it would be nice if you know how to recognize these fermentations. Names originate from the product that is formed. And just as an example, again, you don't have to memorize this pathway, the example of you can have homofermentation and heterofermentation. If product is only one, like in homolactic or homofermentation, then we talk about homofermentation. The other type of homofermentation can be fermentation that produces ethanol, but that happens only if you have ethanol dehydrogenase. So basically, glucose is here substrate that is being fermented, and you see how ATP is spent. And then in this point, fructose 1,6-bisphosphate gets split by aldolase. This is important for the next one. Onto the 2 3-carbon compounds, and each of these 2 3-carbon compounds will go to this process here, which will generate NADH and 2 ATPs. Now, and you end up with pyruvate. So what will happen now, because you want to keep this process going, you have to somehow get back this NAD plus, and you already form NADH. So in order to get second round, you have to get NAD plus. So that is why you have to basically reduce pyruvate, which generates lactate, and NADH is oxidized to NAD plus, and process can continue. In heterofermentation, there are multiple products, not only lactate or ethanol. The difference is that there will be no that aldolase that will create 2 3-carbon compounds. So it has to take, bacteria has to take another pathway. So it goes from 6 molecules of carbon to 5 molecules of carbon, and then it ends up to be split on 1 3-carbon molecule and 1 2-carbon molecule, because there are now 5. And then both of them will go to slightly different process, which will result to one with lactate, the other one with ethanol, but you will also have carbon dioxide as a byproduct. And these lactic acid bacteria help in production of cheese and milk, so they can transform lactose, fat, and protein, and they are actually those that give cheese the flavor, because it's very low pH when lactic acid is produced. That prevents growth of other microbes. So homofermentation is important to get that mild flavor and acidity of cheddar cheese. And heterofermentation is important for these holes in cheese, because if you remember, in heterofermentation, one of byproduct is carbon dioxide, and that gas actually creates these holes. And Another fermentation is butyric acid, very complicated one. It's performed by Clostridium, a famous aerotolerant gram-positive organism. Basically, early fermentation products here are acetate and butyrate, and these products lower pH, and when pH gets lower, it will stimulate genes that will redirect fermentation, and microbes will make butanol and acetone. And last but not least is strict land fermentation, or how I heard today, sticky land fermentation, very nice way to memorize it. So this is a fermentation of amino acids. So usually you have two amino acids, one donor, one acceptor. Alanine and glycine are example. And what you need for this fermentation is to have two amino acids and ADP and PI. And what will be products are ATP, carbon dioxide, and acetate and ammonia. Now, this Strickland fermentation reaction is happening in our mouth. So basically what you have is proline in your saliva, and in plaque bacteria that are usually peptostreptococci. This is important, I would like you to memorize. These peptostreptococci complete glucose fermentation and they make free amino acids. So Strickland reaction is happening between free proline that we have in mouth and amino acids that are made by plaque bacteria. So this fermentation results, of course, in ATP and CO2 production, but also produce this valeric acid. And valeric acid is degraded to ammonia. So ammonia is buffering compound that removes hydrogen ions and reduces acidity in your mouth, which causes dental caries, basically alkalinity of your mouth. And this is pretty much all what I had to say for today. So very short lecture. And I will make sure to reduce it because I always want to have some time for your questions and for discussions and for reviews. I hope you enjoyed and see you next week. Have a nice weekend.